última jornada del ciclo 2020 de Ciencias del Cerebro. En este momento tengo el placer también de presentarles a Elaine Delbel, que bueno, la mayoría de ustedes lo conocerán, pero recordamos un poco su historia, su CV. Voy a pasar al inglés en este momento porque Elaine va a hacer la presentación en inglés, así que hacemos también su presentación en este idioma. Elaine Delbel obtained her PhD at the University of Sao Paulo in 1988 and worked as a postdoctoral fellow in the Physiological Science Department at the University of Manchester. She was appointed as an assistant professor of the Department of Physiology at Dental School of Ribeirão Preto, USP, in 1995. Since 2013, she was a full professor at the same department, and she has been a member of the International Brain Research Organization visiting lecturer team. In 2018, Elaine was recipient of the Bernice Grafstein Award for outstanding accomplishments in mentoring from the Society of Neuroscience. The prize recognized individuals dedicated to advancing women in neuroscience through education. Now she is president of FALAN. FALAN is the Federation of the Latin American and Caribbean Societies for Neuroscience. She is also president of the Neurotoxicity Society. In 2020, she was elected a member of ACAL, the la Academia de Ciencias de América Latina. She is most interested in unveiling new mechanisms involved in the pathophysiology of the neurodegenerative and mental disorders, such as Parkinson's disease and schizophrenia. So, Elaine, thank you very much for accepting the invitation. And now it's time for you to share your screen and start your presentation. Thank you very much, Elaine.
Gracias a usted, Marita, por la invitación portuñol en 2 minutos. Estoy muy contenta, gran honor estar aquí hoy y muchas gracias también a las personas que en lunes a las 9 de la mañana están asistiendo a una presentación. Mi español es horrible, pero hablo, me comunico. Pero voy a intentar hablar en inglés, ¿está bien? Voy a hablar en inglés y espero que a ustedes les guste y si tienen preguntas, mismo durante la presentación, escriban a Marita y puedo contestar cuanto quieran. Muy bien, vamos. So today, I'm talking about, I'm telling you a little history about why we are talking of antibiotics for the treatment of Parkinson's disease and L-DOPA-induced dyskinesia. Let's see. Okay. Let me just tell you what I'm talking today. First, I will explain the history or why we are working with this preclinical model of PD and L-DOPA, why we direct our analysis for neuroinflammation, and why are we talking about dyskinesia and antibiotics. Okay, so first of all, let's look this picture that came this year in Lancet, images of Parkinson's disease. This image is from 1886. It's very old and really have the idea of Parkinson's disease for just old people, but like here, no? But nowadays, it may happen in young age. We have the example of Michael J. Fox that was very young when it starts, but many more people had started this disease, you know. So we have also this, let's say, intermediate Parkinson's disease, that in this intermediate, the patient needs treatment with the case L-DOPA, no, but this treatment is not forever. It's very important for motor symptoms, but something happens in the when the patient are receiving this treatment. I'm gonna tell you about that. And so the end, no, when the proeminent motor and non-motor characteristics are appearing, and so the person is really in a difficult situation. Okay, so Parkinson's for old, but for young, and different symptoms. Okay. I want to make a very a short history because we have been studying the nitric oxide system for many years. And in the end, we had some idea, and we have several publications concerning nitric oxide and motor behavior, okay? So there is an effect, even the inhibitors of nitric oxide synthase, and also the nitric oxide, if is released or increase or decrease in the central nervous system. But we knew from the literature that there is an interaction with NMDA receptors. That is, it means increase NMDA receptor function may increase the nitric oxide release. And so it's difficult to have a view, to review if it's going to be neurotoxic or neuroprotector. Until now, these two actions are allowed or happens for nitric oxide, okay? But I heard this once from John Nichols, anatomy is destiny. And in a class that he was giving, and I love this phrase, and I'm gonna tell you why. Why? Because I am a very visual person. I like to write with my hand, and I like to see images. And I start to work with immunocytochemistry, looking at nitric oxide synthase and tyrosine hydroxylase. This is a picture of, here you have in green, no, tyrosine hydroxylase. And as you can see, both in red nitric oxide synthase, neuronal one, that they are really close, and it's full of fibers of nitric oxide synthase in the substantia nigra. When you look at striatum, you don't see many neurons that are positive for nitric oxide synthase, but there are several ones. And look what happens with the tyrosine hydroxylase terminals. They are really close, less than 2 micrometers, the distance between the fibers and the neurons. So there is we discuss in this publication the possibility of interaction, communication between the two neurotransmitters. And you can see here in the, here you have caudate and of human brain, and this is the NADPH diaphorase. It's the nitric oxide synthase has got this kind of activation of enzyme activity, no, diaphorase. And I must tell you, I was in love with the pictures. I was in love with the relation with the cells and so on. And besides the function, the motor function, we did the question if nitric oxide synthase inhibitor could have an effect in dopaminergic lesion. And we know, I'm sorry about that, that Parkinson's disease, in Parkinson's disease, we have a degeneration of the neurons in substantia nigra, and the neurons are the dopaminergic neurons. So our prediction was if we had an inhibition of nitric oxide synthesis, maybe we could have a protective action. So we use in our lab the 6-hydroxydopamine lesion. This the results I'm going to show you today, it's the microinjection of in the striatum, no? Some in rats, you are going to see we do in the medial forebrain bundle, but in this model specifically, it was in the striatum. And some time after the microinjection, we have, you know, a diminution of the tyrosine hydroxylase neurons and substantia nigra compacta. You can see here the contralateral one, because we do just a unilateral lesion that is perfect, substantia nigra compacta and the ventral tegmental area. Okay, so we were able to have a model of Parkinsonism, may I say. Okay. Good. So that was the work of a student of mine, Margaret Gomes, a long time ago. And as you can see, here you have just the tyrosine hydroxylase immunocytochemistry. And she was, because the way she injected in the striatum, she had a partial lesion of a partial lesion, very light lesion over the striatum, okay? And when she gave the inhibitor of nitric oxide synthase, what happens was that the size of the lesion in the medium striatum and putamen, and also in the fibers of tyrosine hydroxylase fibers in the pallidus decreased. So nitric oxide synthesis inhibition decreased the 6-hydroxydopamine lesion. That was really nice, and without any motor effect. Wow! So we start having something evidence of nitric oxide in Parkinson's. Okay. L-DOPA is the gold standard for Parkinson's disease therapy until now, okay? But treatment improves only motor function. And chronically, L-DOPA treatment may induce in 5 years after treatment, doesn't matter the concentration of L-DOPA, induces this kind of, L-DOPA-induced dyskinesia. This is really a problem, because after you have the first episode, you are always it is always going to appear, doesn't mind if you decrease or stop giving. When the person receives the L-DOPA, it is coming if you have a first reaction. So until now, there is really a great need, and there is a lot of investigations concerning drugs that can modify this dyskinesia, this behavior, okay? Also, always it's important to think about drugs, doesn't matter new or old ones, that can slow or prevent the progression of the disease. It's not an easy disease, we know, okay? So the next question we did, because with nitric oxide, was if it is possible to protect the L-DOPA-induced dyskinesia. And what was beautiful was that our first experiment, just to look nitric oxide synthase, as you can see, there is an increase in the nitric oxide synthase in the striatum of L-DOPA-induced dyskinesia, okay? and also an increase of guanylate cyclase, soluble one. That is another enzyme that is present, that works, that is part of the function of the nitric oxide system. Okay, so we had a nice evidence that something is happening with this system in the striatum and in the brain of Parkinsonian rats with dyskinesia. Okay, so this model was described in 1988 for rodents. It was a great advance because before that it was just in monkeys that would be possible to study dyskinesia. And it's just the lesion. We wait, 20 days until the establishment of the lesion, and after you start a chronic treatment with L-DOPA. And more or less, what happens, it's a dyskinesia of the member, okay? Also a torsion in the body, and also, pardon, and also there is a facial in the face, okay? Sometimes with it's not coming, but doesn't matter. Okay, so the dyskinesia has got 3 kinds at least of abnormal involuntary movements: the limb, the body, and the tongue. Okay, so there is a scale. It goes from 1 to 4, and it's possible to have values, to have a quantification of the intensity of dyskinesia in the animals. It's possible to do that in rats and in mice. Okay, good. So that was the work of my student, former student, today Fernando Padovan. And what he did was he tested at least 2 nitric oxide synthase inhibitor. Well, I'm showing you the action of 7-nitroindazole. 7-nitroindazole in vitro has got behaves like a specific inhibitor of neuronal nitric oxide synthase. You can see here that when you give 10 milligrams to 10 milligrams by kilogram of 7-nitroindazole, it didn't change very much the 3 kinds of dyskinesia. But when we increase the concentration, as you can see, L-DOPA 30 milligrams. Oh, I'm silly, pardon. This is the L-DOPA concentration with 10 milligrams in our animals because let me tell you, we gave the L-DOPA by oral application, okay? And because we want to have the same model as patients. And what happens is that the same problems with absorption that happens with patients happens with the animals. That was the reason we needed to use 30 milligrams. But when we gave it, we have a strong dyskinesia, L-DOPA dyskinesia. That is, it appears here, okay? And when we did dose response concentration of 7-nitroindazole, and you can see that with 3, 10, or 30 milligrams by kilogram, you have, you decrease you completely L-DOPA-induced dyskinesia, no? And what was really important, this when you have, the animal has got dyskinesia, it is not possible for him to do well in this test that is the road performance, okay? So when we gave 7-nitroindazole, 10 and 30 milligrams by kilogram, we have a decrease in the dyskinesia, and also we kept the including it increased the motor effect of L-DOPA. This is the locomotion effect. I'm not discussing it with you now. The important thing this moment is these three parameters of L-DOPA-induced dyskinesia, and it was published in Neuroscience. It was very nice for us. I would like to say that today we have results not just of our lab, the neuronal, the endothelial, and induced nitric oxide synthase decreasing L-DOPA-induced dyskinesia, and also this model of aphakia mice. Okay. Okay, so the literature presents that the non-steroidal anti-inflammatory drugs may be used for Parkinson's disease. We cannot forget that nitric oxide is very active in inflammation. Let's remember the induced nitric oxide synthase, no? So our aim was to look at the neuroinflammation in the brain of the animals with L-DOPA-induced dyskinesia. And it's interesting, and I'm going to show it, that in the human brain, it is demonstrated in alive when they do a scanning in patients, and also in the post-mortem tissue, that there is evidence of a inflammatory process. Okay, so our question, is there an effect of nitric oxide inhibitor in inflammation? And it is interesting that after we start our work, we found this paper from Barnum et al. 2008. In this paper, he showed that external corticosterone decrease lead and L-DOPA-induced dyskinesia, I beg your pardon, and it was dose dependent. And he showed that there is an increase in the IL-1 beta expression in the striatum, and corticosterone decrease this release the expression of IL-1 beta. So our idea, our hypothesis was in the right way. And what we did was to determine the presence of first the astrocytes using GFAP immunoreactivity in the striatum. You can see here that, look, the increase of GFAP was incredible, and the astrocytes have characteristics of activated ones. And in this cartoon, you can see that the expression of astrocytes were really, really intense around capillaries, okay? So that is the control, and it was, it was decreased by 7-nitroindazole. I would like to not just to save some time, to say that microglia using the OX42 expression was increased also, and we verified the induced nitric oxide synthase. So microglia, astrocytes, activated ones, both of them, and the induced nitric oxide synthase was increased. What tell us about an inflammatory reaction in the striatum of the animals with dyskinesia. It was nice. And finally, we saw also the increase of COX-2, the enzyme COX-2 expression in the striatum interneurons of animals with L-DOPA-induced dyskinesia. It is interesting because the COX-2 appeared in the neurons and not in microglia or astrocytes. So we did several studies, and I'm confident that this expression was in neurons, and specifically in some interneurons of the striatum. Okay, and the action of 7-nitroindazole decrease the dyskinesia and the COX-2 expression. Marisa Bortolanza was a postdoc in the lab, and she was the one that analyzed all these results together with Fernando. So we have this evidence of neuroinflammation, and so we have our hypothesis was really, let's say, not completely proved, but evidence that it's true. And that now we think we have like a pro-inflammatory environment in this triad. It is generated by the increase of glutamate release from the cortex, the external dopamine that is produced by L-DOPA. It, you know, may and for the the third factor is the death of dopaminergic neurons in the of substantia nigra, and consequently a decrease in the terminals in the striatum. So these three components would increase the astrocyte, microglia, and mediators of inflammation in the striatum. Good. So I told you about that, but what happens with human brains or patients? What is inflammation about in human brains? You can see here the magnetic resonance where they determine dopaminergic terminals here and the microglia. You can see the putamen here, and the red is the intensity of the ligand of a dopamine transporter, and in that is control, no? And the Parkinson's disease patient, there is a decrease. It means the neurons or the terminals in the putamen are decreasing. We believe, no, the authors believe for because of the Parkinson's disease. And also there is an increase in the microglia proliferation, as you can see here, in the brains of patients with Parkinson's disease. Okay, many studies showing this in patients. I asked, and you would ask to me, and what about presence or not of dyskinesia? No analysis of dyskinesia in those patients, okay? And most of the studies with human brains, no determination of if these dopaminergic terminals decrease or the microglia increase mostly was related with dyskinesia. Okay, so doesn't matter, we go on, we went further. And I'm gonna tell you very quick history about this antibiotic. This happened in Germany, in the Institute Max Planck in Göttingen. So under supervision or in collaboration with Professor Walter Stummer there. Okay, it was an accident. What was the accident? The accident was that my former student, Marcio Lazzarini, he was there working. He was doing his postdoc research, and he did the surgery, doing the microinjection of 6-hydroxydopamine in the striatum. Okay, 20 days after he came, he did the surgery in 40 animals, 40 mice. And after 21 days, when he tried to analyze if there was a lesion or not, what happens was that just 2 of the 40 animals had the lesion. So a big problem, what was happening, no? And so it was a minor mistake, an accident, and something magic happens. What was it? Marcio used the food that was prepared for transgenic animals. So the food had doxycycline, okay? Let's see. So lesion, the food that had doxycycline, and what happens was that the animals didn't present any rotation by the amphetamine test. So we repeat the experiment, not just with the food in the food, because we need to be confident that it was doxycycline. And so he did a group of animals that receive doxycycline subcutaneously, okay? And it happened again, okay? Doxycycline in the shoe or subcutaneously decrease rotations induced by amphetamine in mice when compared to the rotations induced in the animals that receive normal food. Wow. And so you can see here, it is a partial lesion. No, those are the controls, but in substantia nigra also, but in the animals receiving doxycycline, this one in food and here in subcutaneously, no a decrease in the size of the lesion. And also we had a decrease in the, here is, I don't remember, I think it is microglia, I believe so. In the, I don't know if it's microglia or astrocytes, I forgot, I'm sorry, I'm too nervous with these things, but doesn't matter. You can see a decrease with shoe and with subcutaneous, also in the substantia nigra reticulata. Look at that, okay? So we had a nice observation. We didn't throw away all the results, and we confirmed that doxycycline was able to decrease TH. It was able also to decrease astrocyte and microglia expression. And our question was, it was an anti-inflammatory effect. This neuroprotection happened because it was a neuroinflammatory action. Effect of, we came back to our hypothesis, no, concerning this environmental in the striatum, inflammatory, and more or less in this, during this time, 2016, not long after we published the results with doxycycline, we saw this paper. It was done in Denmark, and what happens, the observation was normal people has got, let's say, think 3 of incidence of Parkinson's disease. If a patient has rosacea, it goes to 7, almost 8, the probability of incidence of Parkinson's disease. But the treatment for rosacea is doxycycline. And so they published this study. It was a cohort study, a lot of patients, and they showed that patients with rosacea receiving a tetracycline, doesn't matter if it's doxycycline or minocycline, they had a real decrease of the probability of presenting Parkinson's disease. So it seems we were in our right way, okay? But what was the mechanism, how doxycycline was acting? And we did it together with our partner in France, Rita Heisman-Vosari, and it was some studies using microglial cell in culture, primary culture, and they induced there the activation of, it was Brazilian students that went to France to do that. So with LPS, they increased the activation of microglia, and doxycycline dose dependently decreased the incidence or the activation of microglia by LPS. Doxycycline also decreased the metalloproteinase 3 and metalloproteinase 9 activity in this microglia, and also several cytokines and MAP kinase and NF-kappa B signaling pathways. So in microglia, it was a very nice, important effect of decreasing factors that of inflammation. No, we are doing a research with a group in Ribeirão Preto, and as you can see, his hypothesis was concerning the increase of the neuritogenesis, so the neurites. So those are the control neurons, SH neurons, and here when he gave neural growth factor, you can see it's not brilliant, but there is the neurite growing. And when he gave to but in the culture, the inhibitor of receptor, the antagonist of one receptor of nerve growth factor, it much less neurites. And doxycycline, it was incredible, but also increased more than this concentration of nerve growth factor. No, the dendrites, the neurites, okay? And doxycycline plus the inhibitor of the blocker, the antagonist of NGF receptor also present a decrease. So also he was looking at another system, MAP, ERK, and P3K-AKT, and both of them also are influenced, are part of this effect of doxycycline. Good. So the neuritogenesis, that is something important for neuroprotection. But I didn't tell about that. But for example, doxycycline is an antibiotic from the group of tetracyclines. And many, you can ask, or people can say, wow, what is it possible to give chronically doxycycline to patients? Is it safe and the tolerance of antibiotics? But a group in United States, Golub, it was the name, surname of the research, worked a lot about with chemically modified tetracycline. And the most active, the most safe, the most was this one that's called the CMT3. This drug have a really reduced antibiotic activity, but keeps the anti-inflammatory and anti-angiogenic effect. Wow. So we could have, you know, a molecule that could work like doxycycline, but without the antibiotic effect. And this study was done in France, also in neuron culture, primary culture, and the manuscript is in preparation. As you can see here, they induced the LPS reaction, inflammatory reaction in neurons. You can see that there is an increase when they put LPS, and COL3 does dependent decrease the concentration of TNF-alpha. They showed the same for doxycycline, and the positive control was dexamethasone. And also in the culture, the neurons in culture, they use alpha-synuclein, that is the protein that's present in the Lewy body, no, in the brain of Parkinson's disease patient. And you can see COL3 decreased the alpha-synuclein, the alpha-synuclein induced increase in TNF-alpha. The same thing happened with the antibody against glial cells, microglia, in the presence of inflammation induced by LPS or alpha-synuclein. And the COL3 decrease the IBA1 activation induced by LPS or by alpha-synuclein. The same thing happened with doxycycline and dexamethasone. So we have a very nice effect of COL3 also in neurons. And COL3 and doxycycline decrease inflammation in neurons in culture, primary neuronal culture, and also primary glial culture and cells as age. Good. One thing more, that was a collaboration with Rita in Paris and Rosana Shein. She's from the University of Tucumán in Argentina. She's a biophysicist, okay? And she did the hard work here. And some of the work was done also in France with Rita. Here you have, you know, the Lewy body here, no? Stained in brown. And this is a hallmark of Parkinson's disease, okay? And it is an immunocytochemistry, and it's the presence of alpha-synuclein here. And so they did the question, alpha-synuclein is able to decrease the aggregation, pardon, not alpha-synuclein. Doxycycline is able to decrease the aggregation of alpha-synuclein. They did this study in vitro. And as you can see here, if you have in these conditions that they have as standard, alpha-synuclein, 48 hours, you have some oligomers. If you leave 120 hours, you have the fibrils. But in the presence of doxycycline, no aggregation. It was interesting because, you know, doxycycline even induced the oligomers to go for a non-toxic pathway, non-seeding species. So if you want to have more details, we published this study some years ago, and now this year just went out the effect of the COL3 also, decreasing the aggregation of alpha-synuclein. What is important, no, if we think about human studies. Okay, but now let's see, let's make just a resume what we have. In microglial cell culture, we have inhibition of metalloproteinase, ROS, cytokines and these pathways on FKB and MAP kinase. In neurons in culture, we have a decrease of TNF-alpha, IBO1, ROS, PC12 cells, a decrease of neuroitogenesis, and a decrease of oligomeric species aggregation. Good. So it is a very, the mechanism of action in vitro was well fundamented, okay? And maybe it could be used for preventing or delay the progression of Parkinson's disease. That was the idea since the beginning, no? But we did the same question. Those drugs decrease L-DOPA-induced dyskinesia, and I can show it here. You have L-DOPA treatment in days. Here you have the some of the three kinds of dyskinesia. So it starts very high, but if you give Doxycycline 30 minutes before L-DOPA, it decreases completely, almost completely, the L-DOPA-induced dyskinesia. You can see here, because the measurement happens every 20 minutes until 108 minutes after L-DOPA treatment. Those are the animals that receive Doxycycline, and here just L-DOPA, okay? And one important thing was that the effect in the, we use two motor tests, the stepping test that is important for measurement the capacity of walking in animals and the distance that they travel. You can see here that animals, this stepping test is decreased, no, in 6-hydroxydopamine animals that even with Doxycycline, when they receive L-DOPA, it goes almost closer to normal, and the Doxycycline kept the effect of L-DOPA. The same thing here with distance travel. We hope our paper is, let's say, accepted in British Journal of Pharmacology, and it's coming now. Marisa was Bortolanza was the one that did most of the experiment. We have a similar effect with COL3, look, and with 100 nanomoles, it was a real decrease. I must confess that COL3, we did the injection in the cerebral ventricle. It is a really expensive drug. It crossed the blood-brain barrier, but in this time we didn't have enough. And so we did the central application, and COL3 also, that is the modified Doxycycline without antibiotic action, decrease the L-DOPA-induced dyskinesia. And we did our classics investigation. No, here you see FosB in the striatum of rats with L-DOPA-induced dyskinesia. There is an increase in FosB, COX-2, astrocytes using GFAP labeling, and OX42 for microglia. Animals receiving Doxycycline 15 minutes before L-DOPA, decrease of L-DOPA in dyskinesia, we have a decrease in the expression of the 3 molecular labels of dyskinesia. It is important for me to show you, to tell you that animals, in this graphic, you can see that animals that received Doxycycline together with L-DOPA, since the first day the animals receive L-DOPA, the animals didn't develop dyskinesia. So the treatment, this simultaneous treatment of L-DOPA and Doxycycline block dyskinesia development. And we analyzed also the increase of ROS, no, and of the metalloproteinase in the striatum of animals with dyskinesia, and you can see here that there is a decrease, and this decrease is correlated with the effect is correlated with L-DOPA and dyskinesia. Okay, so what this study now is showing that doxy and COL3 inhibits the development and also the established dyskinesia without affecting the L-DOPA therapeutic effect. This effect is associated, is correlated with inhibition of glial cells activation, reactive oxygen species production, and activity and expression of metalloproteinase. Okay, so maybe it is an anti-inflammatory effect, not only, but at least partially it is acting in this way, okay? And what is very interesting, and we believe we confirm that doxycycline, besides an antibiotic, is also a multi-target drug that may bring many beneficial effects to neurological disorders. It is important, yes, it is important. And it is possible also to give sub-antibiotic concentrations of doxycycline for treatment. And another thing, Parkinson's disease nowadays, it's called a syndrome, or it means many symptoms and signals that happens together, okay? So maybe a multi-target drug may be important. And doxycycline or COL3 maybe in the near future can be tested to reduce dyskinesia in patients with Parkinson's disease. Okay, I don't know how long I talked, but I would like to say thank you to my, it's not the same, this happened 2 years ago. It was in Christmas time. We had a meeting the lab, a party. Now some people went, some people, new people came, and the ones that directly connect to this paper was Fernando Padovan, Marisa Bortolanza. He's a lecturer in the University of São Paulo. Marisa is in Germany. Mauricio is finishing a postdoc with me, Klaus also, and Celia is the technician. I would like to thank you very much, Professor Dr. Rita Heisman-Vosari, for the collaboration, and Professor Walter Stummer. This accident with L-DOPA, doxycycline, happened in his lab, and he doesn't work with Parkinson's. Now he works, and he gave us he allowed us to do the work, and until now we have a strong collaboration. Thank you very much. I hope I didn't speak too much. And now we are back. No, Marita.
Well, thank you very much, Elaine. It was a great talk. Very like the strawberry over the cake.
I hope so, yes.
Yeah. So now I think it's a good time for questions and comments from the audience. I have some questions that I can post later. Preguntas, comentarios de la audiencia para Elaine.
No,
Cecilia
Hola.
Escorza. Hola, Ceci.
Hola. Hola, Marita. Hola, Elian. ¿Cómo andas?
Bien, ustedes.
No llego a ver bien. No sé si me recuerdas, Eliane, de la pasantía de Elaine. La pasantía
sí.
de video.
Sí.
¿Cómo estás?
Por supuesto, bien, gracias. ¿Y tú?
Muchas gracias por la charla, muy interesante, y los resultados, la verdad que muy interesantes. No puedo dejar pasar la pregunta de, al ser la doxiciclina un antibiótico, ¿ustedes creen que existe la posibilidad de que esta droga esté modificando de alguna manera la microbiota intestinal de los animales y que una parte de su efecto sea a través del eje bidireccional intestino-cerebro, modificando de alguna manera alguna acción central de la droga?
Es posible, pero lo que le digo es que la doxiciclina en Estados Unidos fue liberada. Ellos pueden utilizar la concentración de 40 miligramos por día en humanos para el tratamiento de la rosácea, ¿sí? La infección de la piel. Y también es posible utilizar la concentración no antibiótica, 40 miligramos, para el tratamiento de la periodontitis. Y hay estudios de aplicación, de utilización de la doxiciclina hasta 2 años en la dosis subantibiótica para el tratamiento de rosácea a lo menos la descripción en los papers es que prácticamente no hay efecto en el tracto gastrointestinal. Pero todavía hay que procurar más si hay alguna cosa o no. Porque yo tengo así, es una cuestión todavía. No es que no creo, por supuesto, hay la ligación del intestino por el nervio vago, hay también de la flora bucal, ¿sí? Y, ¿por qué no? Es una posibilidad. Pero hay que estudiar más, ¿tú no piensas? Hay que pensar un poco más sobre esto, ¿no? Sí.
Sí, totalmente. Es que está rompiendo un poco los paradigmas que tenemos los neurocientíficos sobre que las acciones de las drogas son exclusivamente a nivel central. Entonces, por eso te hacía la pregunta. Hay muy poca evidencia todavía, pero no quiere decir que no nos hagamos la pregunta.
Sí.
Yo creo que hay que, buscar en la literatura si alguien hizo doxiciclina en microbiota intestinal, en animales, en tratamientos donde se demuestra que el efecto es antibiótico. También estaría interesante ver una dosis subantibiótica.
Sí, sí.
Y es un trabajo que hay que conseguir un socio microbiólogo para hacer ese estudio. Sí. Pero creo que es eso, es que lo del eje intestino-cerebro está rompiendo un poco los paradigmas y los dogmas que nosotros mantuvimos durante mucho tiempo. Por eso te hacía la pregunta, porque era como, justamente, hacías tanto énfasis en su acción antibiótica, porque las drogas son varias cosas. Capaz que en su acción antibiótica no tiene nada que ver con su acción antibiótica y es, no sé, tiene un mecanismo de acción justamente por lo multitarget que vos hablabas de la sustancia. Pero muy interesante. Sí.
Salió recientemente un review inglés, de un hospital inglés, que ellos estudiaron el efecto en el microbioma de los antibióticos más utilizados en Inglaterra. Y la tetraciclina está allá, creo que es la doxy, nunca me voy a recordar muy bien. Y también, pero como antibiótica, la concentración antibiótica, ¿está bien? En humanos. Y me parece, y por lo que me recuerdo que leí, también son mínimos los efectos gastrointestinales. Pero no es el problema. Con el hombre, cada uno es uno, ¿no? Y cada uno tiene sus características. Yo nunca encontré un estudio profundo y haciendo comparación, ¿me entiende? En animales. Tú tienes razón, pero lo que le digo es que la evidencia en la literatura con la dosis subantibiótica y este review es reciente, es de ahora, 2020, si quieres te puedo enviar después la copia de los papers, ¿sí? Dijo que no, que son mínimos los efectos, pero no se puede decir lo que el efecto mínimo va a causar, ¿no? Entonces es una cosa para pensar, por supuesto. ¿Está bien?
Muchas gracias, Elaine.
Gracias a ti, Cecilia, por tu atención.
¿Alguna otra pregunta, comentario de la audiencia? Bien, bueno, yo tengo un comentario. En realidad tu presentación me llevó a uno de mis primeros amores en las neurociencias, que es la enfermedad de Parkinson y el sistema nigroestriatal. Fue mi trabajo de maestría hace muchísimos años. Y me acuerdo que en aquel momento proponíamos la importancia de la transmisión nicotínica digamos, en el mecanismo subyacente o la neuroprotección frente a mecanismos que podrían estar induciendo la enfermedad de Parkinson, trabajando justamente en el mismo tipo de modelo, ¿no?, que trabajan ustedes con 6-dioxidopamina. Y he encontrado que los receptores nicotínicos participan también en la activación microglial, y a su vez que ese antibiótico afecta la expresión de receptores nicotínicos. Entonces, ahí como que atando el ciclo, mi pregunta, que ya te digo, me vuelve a lo que era mi tesis de maestría hace muchos años atrás, si el efecto del antibiótico no podría estar mediado, neuroprotector, ¿no? No podría ser que esté mediado por su acción a nivel de receptores nicotínicos.
¿Por qué no? Lo que le digo también, no estudié esto directamente, pero lo que le digo es que, por ejemplo, el incremento de la expresión de la COX-2 ocurre en las neuronas colinérgicas. Hay colocalización entre COX-2 y
entonces es muy probable, cierra.
¿Me entienden? Sí. Entonces es muy bonito esto, ¿sí? Y y también ahora es como hace un año hubo un revival of cholinergic transmission. En Parkinson. Sí, hay cosas muy interesantes.
Esto fue por los 90.
Sí, pero después es como la moda y la neurociencia cambia, ¿no? Pero están trabajando muy serio porque hay nuevos resultados, nuevas evidencias con el sistema colinérgico.
Mira tú. Yo no he seguido el tema últimamente porque ahora estoy en otra área, ¿no? Pero es como que son esas cosas que cuando cualquier presentación sobre el sistema nigroestriatal y los ganglios basales, el Parkinson, como que a uno le trae esas cosas, y bueno, en aquel momento proponíamos justamente eso como hipótesis, el rol neuroprotector de la transmisión nicotínica.
Se puede hacer muchos estudios farmacológicos, ¿no? Tanto in vitro cuanto in vivo, ¿no? Intentar desear somación, cosas así, ¿no? Es muy interesante, no se termina estas cosas, ¿no? Hay mucho que hacer, ¿no?
Sí, siempre eso es lo lindo que tiene la investigación, que siempre hay cada trabajo lo que hace es responder, a veces parcialmente, las preguntas que uno se plantea, a veces más o menos completamente, pero siempre abre el camino a nuevas preguntas, ¿no? Y de eso se trata justamente, ¿no? Avanzar el conocimiento gracias a ese ciclo virtuoso de trabajar y que al trabajar se generan nuevas cuestiones, ¿no? Que nos van corriendo un poquitito la frontera del conocimiento. Bueno, Zulma dice: muchas gracias, doctora Castelló, por compartirnos estas sesiones tan académicamente gratificantes. ¡Ay, eso es para mí! Era para él. Debo retirarme y aprovecho para desearles una saludable temporada navideña y fin de año, y gracias. Bueno, encantadísima, Zulma, y bueno, me alegro mucho y disfruto mucho con esas palabras. Este fue justamente un esfuerzo en épocas de pandemia, de tratar de mantenernos activos y unidos en la región. Y bueno, en sintonía con otras cosas que están pasando, que son muy lindas, y que en eso están participando Elaine y otros tantos colegas de la región, como es la creación del proyecto Cerebro Latinoamericano. Y bueno, en ese camino estamos. Así que esto es un granito de arena en ese sentido. Te agradezco enormemente, Elaine, por esta charla que cierra el ciclo, que fue preciosa, y bueno, agradezco a todos los que están presentes y a los que estuvieron en las distintas charlas de todo este ciclo, y ya estamos pensando en el año que viene, así que esténse atentos, que bueno, pronto habrá alguna manera diferente, pero seguirá el seminario de Ciencias del Cerebro tratando de mantener este hilo de conexión, o esta red de conexión para mantener a las neurociencias dentro del Uruguay, en la región y con el mundo en conexión, justamente. Así que bueno.
Gracias a usted, gracias a usted por la invitación y por organizar esto. Muy bien.
Bueno, gracias. Un abrazo a todos y bueno, nos veremos pronto. Felicidades y cuídense. Chao.
Chao.